Good morning, everybody. In our last class, we studied about the different techniques in rDNA technology, the steps or the methods of rDNA technology. In this class, we shall be discussing about the tools of rDNA technology. One tool, namely an enzyme, restriction enzyme or restriction endonucleases. They are commonly called as restriction endonucleases, which are necessary for rDNA technology to cut the DNA. So you must be remembering that I told you in the last class, when we have to cut the plasmid and the gene of our interest, we have to use an enzyme called as restriction endonuclease. So today let us discuss about the enzyme restriction endonuclease. Now, what are restriction enzymes? Restriction enzymes are found in bacteria and other prokaryotes. What do? They recognize and bind to specific sequences of DNA called restriction sites. So there will be some sequences of DNA, some length of DNA, which the restriction enzyme will recognize that length of DNA, and that length of DNA is called as restriction site. The restriction enzyme goes and binds to that restriction site. Each restriction enzyme recognizes just one or few restriction sites. When it finds its target sequence, a restriction enzyme will make a double-stranded cut in the DNA molecule. So DNA is double-stranded. You all know that. So it makes the double-stranded cut. In both the strands, it will make a cut. Typically, the cut is at or near the restriction site. At the restriction site, it may cut the DNA, or somewhere near the restriction site, it may cut the DNA. And the cut occurs in a tidy and predictable manner. So now, why do the bacteria have restriction enzymes? It is because to kill the DNA of their enemies. What are the enemies of the bacterial cells? The bacteriophages. So these phages, when they invade the bacterial cells, the bacteria have these enzymes, restriction enzymes, which cut or cleave the phage DNA and kill it off. So like this. These are the people responsible for the discovery of restriction enzymes: Werner Arber from Switzerland, Daniel Nathans from USA, and Hamilton O. Smith from USA. They got Nobel Peace Prize in Physiology or Medicine in 1978. Now, as an example of how a restriction enzyme recognizes and cuts a DNA sequence, let us consider EcoRI from E. coli. EcoR1. It is a restriction enzyme, a common restriction enzyme which is used in labs from E. coli bacteria. EcoR1 cuts at the following site. See, the double-stranded DNA, one strand runs from 5' to 3' end, another strand from 3' to 5' end. So, this will be having, the 5' to 3' end strand will be having GAATTC. The other strand will be having the same basis in reverse order, CTTAAG. So, this is the EcoR1 site. The enzyme, restriction enzyme, EcoR1 recognizes this specific sequence. So, this is the site which it recognizes. So, when EcoR1 recognizes and cuts this site, it always does so in a very specific pattern that produces ends with single-stranded DNA overhangs. So, how does it cut it? See, it goes and cuts in between G and A in both the strands. In between G and A, here it goes and cuts in between G and A. See, it cuts here in between G and A, and here also in between G and A. So, in this manner it cuts so that it leaves the ends will be single-stranded DNA overhangs. See, here you have one end here which is an overhang, and here one end which is a overhang. So, this is the manner in which the EcoR1 enzyme cuts in its particular restriction site. If another piece of DNA has matching overhangs, for instance, because it has also been cut by EcoR1, the overhangs can stick together by complementary base pairing. For this reason, enzymes that leave single-stranded overhangs are said to produce sticky ends. Like this only, if there are other sticky ends, the DNA will go and bind because there will be complementary base pairing. So, since they go and bind automatically, for this reason, the enzymes that leave single-stranded overhangs are said to produce sticky ends or staggered ends. Sticky ends are helpful in cloning because they hold two pieces of DNA together so that they can be linked by DNA ligase. Not all restriction enzymes produce sticky ends. Some are blunt cutters, which cut straight down the middle of a target sequence and leave no overhang. The restriction enzyme SMA1 is an example of a blunt cutter. So here is the picture of SMA1 enzyme. This is the restriction site which it recognizes, CCCGGG, GGGCCC, and it cuts in between C and G, and it cuts directly straight with no overhangs. It does not leave any overhang. So it is a blunt cutter. It cuts straight. Blunt ended fragments can be joined to each other by DNA ligase. However, blunt-ended fragments are harder to ligate together. The ligation reaction is less efficient and more likely to fail because there are no single-stranded overhangs to hold the DNA molecules in position. So, the enzymes, restriction enzymes, can cut either by producing sticky ends or blunt ends. Now coming to DNA ligase. If you have learned about the DNA replication, you may already have met DNA ligase. In a DNA replication, the ligase's job is to join together fragments of newly synthesized DNA to form a seamless strand. Seamless means continuous strand. The ligases used in DNA cloning do basically the same thing. So same thing, that is joining. So DNA ligase is also called as molecular glue or gum. Restriction enzymes are commonly called as molecular scissors because they cut the DNA strands. These DNA ligases are called as molecular glues because they join the DNA strands. If two pieces of DNA have matching ends, DNA ligase can join them together to make an unbroken molecule. So here, as we can see, sticky ends stick together but gaps remain. The DNA ligase seals the gaps. How does DNA ligase do this? It does it by using ATP as an energy source. Ligase catalyzes a reaction in which the phosphate group sticking off the 5' end of one DNA strand, the phosphate group of one DNA strand which is overhanging is linked to the hydroxyl group sticking off the 3' end of the other. So this is how it joins. So this reaction produces an intact sugar phosphate backbone. So what does the ligase do? The ligase catalyzes a reaction in which the phosphate group sticking off the 5' end of one strand is linked to the hydroxyl group sticking off the 3' end of the other DNA strand. So it in turn produces an intact sugar phosphate backbone. Now building a recombinant plasmid. Let us see how a restriction digestion and ligation can be used to insert a gene into a plasmid. This we have studied in our previous class. Once again, suppose we have a target gene. This is the target gene. This is the target gene flanked with EcoR1 restriction sites. So this is center, light blue is the gene. The purple regions are the EcoR1 restriction site. See, here it's enlarged. This is the EcoR1 restriction site. This is the EcoR1 restriction site, and this in between is our target gene. containing so this is our target gene. Suppose we have a target gene flanked with eco-RI recognition sites, and we also have a plasmid here containing a single eco-RI site. So plasmid also has got one eco-RI site, restriction enzyme recognition site, one eco-RI site. So here we have a target gene, and then we have our plasmid. So our goal is to use the enzyme eco-RI to insert the gene into the plasmid. So our goal, what is our goal, is to use the enzyme eco-R1 to insert the gene into the plasmid. We have to insert this gene into this plasmid, isn't it? First, we separately digest or cut the gene fragment and the plasmid with eco-R1 restriction endonuclease. Okay, first what we do, we separately cut the gene fragment, the gene of interest, and the plasmid with the same restriction endonuclease enzyme, which is eco-R1, the enzyme. This step produces fragments. So we can see here there are fragments, all bits of DNA. Next, we take the gene fragment and the opened up plasmid, linearized plasmid, and combine them along with DNA ligase. The sticky ends of the two fragments stick together. See here, you can see the sticky ends have stuck together by complementary base pairing. Once they are joined by ligase, the fragments become a single piece of unbroken DNA. The target gene has now been inserted into the plasmid, making a recombinant plasmid. So, this is what has happened. What happens? Once they are joined by ligase, the fragments become a single piece of unbroken DNA, and the target gene, which we had isolated, has now been inserted into the plasmid, making a recombinant plasmid. Now, restriction digests and ligations involve many molecules of DNA. So, in this example, we saw one outcome of a ligation between a gene and a plasmid cut with eco-R1. However, other outcomes could happen in this exact same ligation. For example, the cut plasmid could recircularize, close back up without taking the gene of interest, our gene of interest. This can happen. So the second middle diagram, plasmid closes back up without incorporating our gene. Similarly, the gene could go into the plasmid but flipped backwards. The third diagram. So, gene can go into the plasmid, but it is oriented ulta, backwards, since its eco-R1 sticky ends are identical. So, gene goes in backwards. That is also possible. Or the the correct method is the first one, that is the gene goes forward. So this is the one which we want, the tick marked one, that is the one which we want of our interest. So like this, many molecules of DNA can be produced. So restriction digests and ligations like this one are performed using many copies of plasmid and gene DNA. In fact, billions of molecules of DNA are used in a single ligation. These molecules are all bumping into one another and into DNA ligase at random in different ways. So if multiple products can be made like all this, all of them will be made at some frequency, including ones we don't want. So the second one, third one and all we don't want. First one only we want. How can we avoid the bad plasmids? When we transform bacteria with DNA from a ligation, each one takes up a different piece of DNA. We can check the bacteria after transformation and use only the ones with the correct plasmid. So that is a different procedure called selection. In many cases, plasmid from transformed bacteria is analyzed using another restriction digest to see if it contains the right insert in the right orientation. So now these are few examples of the restriction enzymes. So you can see these are different restriction enzymes and their recognition sites and the regions where they cut. So the first two produce blunt ends. The last three produce sticky ends. So, these are some restriction endonucleases. These are the references. Thank you.